It's cute. It's swaggy. It's the super kawaii Desu Swaggie Bio Show. I'm your host, Big Wang, here with Harvard PhD in swag, glory hole, and 2 Chainz University Dr. QUINCY Ho Chi Minh. Today we gonna understand the process of DNA transcription into amino acids. I'ma start this turn up. Before we begin, we gotta get in the right hood. We gotta travel to the nucleus hood. Zoom! I got a bunch of my homies up in here. We're gonna start this off with some DNA, which is comprised of nucleotides such as AA-9, West Cytosine, Guantanamo Bay 9, and Tyrone sign. AA-9 be hooked up with Tyrone sign, and West Cytosine be with Guantanamo Bay 9, which I say is a very abusive relationship. We got multiple hookups and long-distance relationships up in this hood, in the form of non-parallel double helix structure. Why? Cause it got swag. Once in a while, my homies get divorced, and the government helicase, being super protein supremacists, be divorcing my homies. Unzipping them jeans, just like how my girlfriend unzips mine. Ooh! Then they gone paired up with some new hookups and one-night stands from RNA. They promiscuous, girl. These RNA are different though. Whereas the DNA be made up of deoxyribose, RNA be made up of ribose only. Then them DNA nucleotides get paired up with the respective RNA nucleotides by RNA polymerase. RNA polymerase first contacts to the start codon methionine and goes down a divorce line of DNA, making RNA off of the template strand of DNA, having opposite corresponding matches until they reach the stop codon, UAA, where then the RNA polymerase says, "We in the wrong hood." On the other hand, the coding strand be matching with the RNA strand, except RNA has uracilin instead of Tyrone sign. You could say that DNA be pulling up to the scene with its uracilin missing. Ha! 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 Unfortunately, them relationships between RNA and DNA don't last long. After the RNA gets laid with DNA and one-night stands, DNA nucleotides be calling their ex-wives back with the help of topoisomerase. Marriages are formed again by hydrogen bonds, and DNA is back together again. This entire process be called Waka Flocka RNA transcription, son. I'ma let Glory Hole take it from here.
Thanks, Big Wang. Now we will talk about RNA splicing. The result of the divorces and one-night stands between DNA and RNA results in a pre-mRNA strand full of unnecessary lines of demon nucleotides. It needs to be refined before becoming a messenger RNA strand in a nucleotide exorcism. After that disastrous one-night stand, some of them RNA nucleotides didn't clean properly and contracted UTI. They're known as introns, useless nucleotides that aren't necessary for the sequence, usually known as trash. Before becoming mRNA that can be translated, they need to be taken out of the equation by splicing them out of the RNA love line, just like how I splice the guts of my enemies before eating their liver. The rest of the nucleotides that want more meaningful relationships are called exons. With the help of spliceosomes, a complex of ribonuclear proteins, or snRNPs, the introns are taken out and the exons are covalently bonded together, forming a loop of useless introns and a strand of refined mRNA. Then other enzymes add on a 5' cap and a poly A tail at the ends of the refined mRNA. So in translation, ribosomes will know when to stop making babies. The mRNA then leaves the nucleus hood and then enters the cytoplasm. The rest can be taken care by
QUINCY Ho Chi Minh. Thai glory hole, much love in central dogma, much DNA, very transcription. Wow! Now we talk about translation from RNA to amino acids. mRNA is expelled from the nucleus and into the suburban cytoplasm looking for love. They're looking for a daddy to make babies with. That is why they went to a local bar known as ribosome and got drunk. There are 3 parts of the ribosome bar: the exit site, peptidyl site, and the amino acyl site. The mRNA goes through between the large and small subunits and hook up with the tRNA with matching anticodons at the amino acyl site, where Viagra is served and babies, otherwise known as amino acids, are attached to tRNA. Unfortunately, the condom broke. Much sabotage. tRNA moves to the peptidyl site, throwing its baby into a dumpster at the end of the chain of amino acids created from likewise other unsupportive DNA. This is done at the peptidylate site. The sugar daddy leaves the ribosome to make room for other very unsupportive daddy tRNA and exits the exit site of the ribosome. At the end of the mRNA translation, the specific binding of the babies is used to make a huge baby monster full of amino acids, otherwise known as a protein.
That's all the time we have. Remember, if you ever need to translate DNA, unzip, divorce, and make babies. It's cute. It's swaggy. It's the Super Kawaii Desu Swaggie Bio Show!